Okay, so I'd like to thank the organisers for inviting me to give a presentation today. I would like to add I am a basic scientist, and there's been some very exciting presentations given about interruptive visualization of the conduction system. What I'm going to be able to show you today is definitely not interruptive, and everything that has been done on ex vivo human hearts. But hopefully you'll be able to see some of the value that the technique that we've been using in our lab in Manchester has, particularly for like some of the variation that has been discussed in earlier presentations. Okay, so briefly in my talk, I'll give an outline about why anatomical models are important for us to create, and also how we can use the micro-CT to create these anatomical models and how we can visualize the cardiac conduction system. And then I'll briefly talk about two studies, one which we published, one which we're currently working on, where we've used micro-CT and the visualization of the conduction system. And then finally, a potential use for some of the models. And I haven't got any disclosures. So why are anatomical models important? Well, they allow us to have detailed analysis of cardiac anatomy, and they can help enhance our understanding of the cardiac function. They also allow the true extent and the proximity of the cardiac structures to be fully appreciated, where previously histological techniques and things might have missed out bits of tissue and from dissection. I'll show from the micro-CT, everything's intact, so you won't miss out on any potentially interesting features. And so once we've got these models, we can look at how things change with diseases and in aging, as well as in the healthy normal heart. So the anatomical models form an important part for 3D computational models, which can also be used for biophysical simulations. So a heart model for the full simulation will require detailed anatomical information. And the earliest models used for studying the heart often didn't include any cardiac conduction system and were very simplified, quite often just ventricular ellipsoids. But using our anatomical models, we can include information about the cardiac conduction system. We can get the full 3D volumetric meshes of the myocardium and other structures such as the vessels. And we can also get information regarding the myocardial architecture, so the alignment of the myocytes. And this is all important for how the heart is going to function and be stimulated. Along with these, it's important to have electrophysiological models. So if we identify the different cell types within the heart that we're going to have in our model, we can have individual electrophysiological action potential models. So then the heart will have the right cell types and behave in a more natural way. And then we also have biomechanical models, so we can simulate fluid dynamics and contraction models into these. And so the more detailed we can get each of these elements, the more valuable that our simulations can potentially be. And then these models can be used to help study disease, where we'd expect to see some forms of structural remodeling in the myocardium and the conduction system. These will modify these elements. And we also get the electrical remodeling, which then can be incorporated into the electrophysiological models and the biomechanical models. So, as I said, our group is interested in the conduction system, and we've been creating 3D reconstructions of the conduction system for a long time. We start out using histological-based systems and immunohistochemistry and segmenting out serial tissue sections. But for the last almost 10 years now, we've been moved to a micro-CT-based system. So the micro-CT works on standard X-ray kind of imaging properties. So we have differential X-ray attenuation between different tissues to be able to identify them. And the advantage of it is, for us, is that it's a non-destructive visualization of the tissue. And it means, as I said, we're not going to damage any structures that we might not know are lying there, and it also means the tissue is still available to do additional experiments with afterwards. So if we need potential histological verification, then we've still got the tissue. And it also opens up the potential to study hearts held in valuable collections. So there's potentials to study various congenital abnormalities and things that hospitals might hold in their collection. So we could study these and then return them to them in their original state. So for the micro CT, we have our sample, which sits in between the X-ray generator and the X-ray detector, and our sample is just rotated through 360 degrees, and we collect between 2,000 and 4,000 projections. For our whole human hearts, using the systems that we have, we get resolutions between 50 to 100 microns, depending on the size of the hearts. So some of the hypertrophied hearts, the resolution will be a little bit lower, but this is what we're currently working with, and we have got working on steps to try and reduce the resolution that we can go down to. As I said, we rely on the attenuation difference to identify our tissues, and so for this we can use various contrast agents, and the main one that we use for looking at general myocardium and the conduction system is an iodine-based contrast agent. And the hearts are basically just immersed into this contrast agent for potentially up to 3 weeks to allow diffusion through the myocardium, depending what features we're particularly interested in seeing with the scan. And on the left here, we have a heart that was scanned without a contrast agent. So if you look closely, it might not be that clear, but there's like some differences between like the fibrous tissue that you can see from the myocardium. On the right is a heart with a contrast medium use, so we can see a lot of the internal details. So once we've got the CT data, we can use back-filtered projection to create the tomograms, so we can see in great detail the internal structures of the heart. And once the CT data is generated as well, we can explore it in any way that we want to. So we can section the hearts like virtually in every plane. So it's very useful for identifying all the features. So we can identify landmarks for the conduction system. We can trace out coronary vessels. We can look at the aortic route and outflow tracts. So it offers a great value to explore the hearts. And they also allow volume rendering, and then the hearts can be also explored by traveling into them. So you get a good view of how structures are related to each other, which can be very useful. And we can also use virtual reality imaging. So we can have segmented hearts that can be explored freely with virtual blackboards to show the features of interest, which can potentially be used for educational purposes. And we also have potential to use with, this is looking at Microsoft HoloLens. This is almost like an augmented reality with see-through glasses. So you can have models within rooms or potentially within like operating theatres or things, if this could be incorporated. Okay, so as I said, as a group, we're interested in the cardiac conduction system, which are the myocytes involved in the generation and conduction of the cardiac impulse. And so we're interested in all components, so the sinus node, the atrial conduction axis, and the His bundle Purkinje fiber network. So this is classical histology looking through the sinus node, which is the pacemaker of the heart. It's located in the right atrium at the junction with the superior vena cava, and it extends along the intercaval region. And in this mass and trichrome histology, we have myocytes in red and the fibrous tissue in the blue. And so we can see here that we have this discrete sinus node area, which is full of fibrous tissue, punctuated by the sinus node artery, and it's lying on the subepicardial region. We can also see how, compared to the atrial myocytes, that the sinus node myocytes are smaller, and they also stain more palely with the masum trichrome. So here we have a micro CT through a tissue block with the intercaval region, and this is the micro CT as we slice through perpendicular to the crista terminalis, as we would do if we were doing traditional histology. And we can see this area in between the atrial muscle and going towards the superior vena cava, where there's this area of low attenuation. And so with the CT, we're able to segment out this area through each of our CT blocks. And when this is compared to what we see in traditional histology, we can see that this area of low attenuation that we're seeing in the CT matches up very well with where we'd expect to see the sinus node located. So this was helping to support our thinking that what we were seeing with this area of low attenuation was actually the area of the sinus node. And so then we're able to do segmentation. So here we just segmented the atrial myocardium, and we've got the sinus node segmented along with the sinus node artery running through it. So using this technique, we can get detailed measurements of the sinus node. We can try and see if there's any projections coming out of it. And here we've used, in this one, we've used a different contrast agent, but this time we've segmented out the fatty tissue from the tissue block. And so we're able to pick up all the fat, and this is potentially useful for, particularly if you're studying obesity or in the aging, where we might expect to see fatty deposition. So we could see how this is infiltrating into the tissue blocks easily. And this is all things that would impact on the activation of the atrial myocardium by the sinus node. So then we move on to the atrioventricular conduction axis, which forms the sole direct myocardial connection between the atrium and ventricles in the healthy heart. And this is found in the lower posterior region of the right atrium. And as has been mentioned before, the anatomical landmarks of the triangle Koch are often used to identify the location of the AV conduction axis. So we have the tendon of Tadaro, tricuspid valve annulus, and the coronary sinus ostium. And so the components of the AV conduction axis, major ones are the compact node, which is like the true AV node, which is situated on the right side of the atrioventricular muscular septum. And when we perform histology, looking at the AV junction, it's seen as histologically dense packed structure, and it stains more palely compared to the surrounding myocardium. And it's separated from the ventricular myocardium via the central fibrous body. And it has connections going to the atrial myocardium via loosely packed transitional cells, as we can see up here. So it's been shown that there's inferior nodal extensions that can extend from the compact node towards the tricuspid valve annulus, and these are thought to act as input pathways into the AV node. And then we move going down more towards the ventricular side. We get the penetrating bundle, which forms when the compact node starts to form through the central fibrous body. And then this is completely insulated from the working myocardium. And then as we go down towards the ventricular septum, the penetrating bundle starts to branch into the left and right bundle branches, which will then go on to connect with the Purkinje fiber networks in the ventricles, which then will form the terminal junctions with the working myocardium. So similar to what we did for the sinus node, we've got the scanned AV node block. And then this is the CT as we're taking sections through this block virtually, and we can see how the structure moves through the central fibrous body, as we'd expect to see from the histology. So what we're seeing on the CT seemed to match up with what we'd see histologically, and when we did histology, we can see very, very clearly that the areas that we'd identify histologically correspond exactly with what we see in the CT. So again, this supports our use of using the micro CT for studying the cardiac conduction system in this way. And then this is just tracing back to the original tissue block and the triangle cock, and where our actual reconstructed AV node is situated. So we can see that indeed in this heart, the AV node is at the apex of this triangle. And we're also able to segment out the AV node or artery supplying the AV node here. So again, once we've got all this information, we can segment it any way that we want. So in this tissue block, we've segmented out the atrial myocardium, the ventricular myocardium, we've got valve leaflets, we've got the aorta segmented, and we've got the cardiac conduction system. So on top of this, we can have the coronary vasculature, and so all of this could be useful for a computer modeling to identify all the complex structures that are going on in these regions. And also, as we mentioned, like with great variation, potential like multiple nodes and things, like this could be very useful to have actual models, and we could perform computational simulations of how this might affect conduction through this area, and then potential like model like therapies of how like different interventions might affect what's going on here. And there's also a great amount of interest because of like the introductions of his pacing and things. So there's lots of interest trying to get the AV conduction axis mapped and modeled in a detailed way. So I'm just going to show the first study where we used the micro CT, and this was in creating a model of the cardiac anatomy in a whole human heart. And this was published in 2017. And this study just involved healthy adult female hearts. And so the whole heart was scanned with the iodine contrast agent, and based on the attenuation differences that I've shown before, we're able to see the sinus node located in the intercaval region. And in this heart, the sinus node length that we saw was around 1.5 centimeters. And this corresponded well with what we'd seen in other studies and in other hearts that we've looked at histologically in the lab. And also in this heart, we were able to see some projections coming out of the sinus node going into the atrial myocardium. You can see some parts here, and we're also towards the inferior vena cava, we're able to see some small islands, which also had low attenuation. And we've seen in some of our immunohistochemical studies in animals that you can get pockets of pacemaking cells down towards the inferior vena cava, and these could potentially act as subsidiary atrial pacemakers, particularly when the sinus node becomes diseased and isn't functioning. So then moving on to the AV conduction axis, we're able to see that the AV conduction axis had a attenuation property that was in the middle of the low attenuation of the fibrous tissue and the high attenuation of the working myocardium, and we're able to extract the AV conduction axis, and it was very similar to what we were hoping it, or we were predicting it would look like. And then we're also able to segment out the His bundles. So we had the left and right bundle branches, and we could see them running on the endocardial surface, and the right bundle branch, as we would have expected to see, was more of a cable-like structure, whereas the left bundle branch was more of a fine fan-like structure. And then we got the Purkinje networks, which were traversing the ventricular chamber and undergoing numerous ramifications. We didn't get all of the interconnections between Purkinje fibers. And so these can all be incorporated into one view, so we can see the interrelationship between all the structures, and then we can also add the extra structures. So we've got the segmented myocardium, so we can perform activation studies on these, and we've got the conduction system, and then finally we can add the coronary vessels, so we can potentially see if, like, there's infarcts, we could see how this might affect this heart, and then this could potentially be included into a computational model. So as I mentioned earlier, one of the important things to try and have in a computational model is the actual myocardial arrangement, and we've been able to extract this from using our high resolution micro CT scans. So here we've used eigenanalysis of the 3D structure tensor, and these are the sections taken across the left ventricle. So these are short axis sections, and so we can see as we cross the ventricular septum, there's a helical arrangement of myocytes, and there's the central zone is more circumferential than the extremities. And on the free wall side, we see a similar pattern, but it's not as distinct as we see in the septal side, and it's consistent from the base to the apex. So now, just touching on some of the current work that we're looking at, and this is using the micro CT, looking at the obese human heart. So obesity is a known risk factor for developing heart disease, particularly with conduction defects. And so what we've obtained is the micro CT data sets from 3 hearts. We've got one with no known cardiac defects. Then we've got one with a high BMI, a reduced ejection fraction, and one with a high BMI and a preserved ejection fraction. And so just from looking at the hearts, we could see the obese hearts were much larger than the normal heart. But what we could see is that with the heart with the reduced ejection fraction, we're seeing some evidence of dilated cardiomyopathy with severely dilated ventricular walls and dilated ventricular chambers. And we could see when we take measurements of the thickness, it's the LV was down to almost like a third of in the normal heart. But in the heart with the preserved ejection fraction, we saw evidence of hypertrophy in the ventricular walls. So then we wanted to look at the conduction system. So these are, here we're looking at the right atrium. And so we're looking at the crista terminalis in the middle. We segmented the sinus node, and we didn't see any like striking differences between the areas of where the nodal tissue was. And when we've taken measurements, there's not great differences, but actually we're wondering whether the size of the sinus node could be associated with the size of the heart. But actually, the largest sinus node was seen in the smallest normal heart. We need to do a bit more investigation into this, but there is potential of whether, as I mentioned earlier, if we're getting fatty infiltration into the area of the node or anything like that, whether that's going to affect what we're seeing on the CT. So it's something we need to try and confirm. Somebody looks at the AV conduction axis. So here we saw in the normal heart, we saw the conduction axis as we'd expect to see with cable-like right bundle branch and a bit of a fan-like left bundle branch. But in the obese hearts, the bundle branches appeared to be much more diffuse structures. So in the segmentation, they appear larger, but we're thinking whether this is also down to the area becoming less specific of being with AV nodal myocytes, and whether we're getting some kind of infiltration of fat into there or increase in fibrotic cells. So again, this is something we need to try and confirm what's actually changing in this area. And then we've also done some work on the coronary vessels. So we see some evidence of revascularization in the obese hearts with a large number of like small vessels as we go to the periphery. So briefly, I'll just touch on a potential use for like these computational models, and this is in transcatheter valve implantation. So someone probably knows that it's done to replace the native valve when it's diseased, particularly in the treatment of aortic stenosis for high-risk patients. There's a couple of valve types. So there's either like self-expanding or balloon expanding, but the principle's the same, that the valve is passed in the aorta and then expanded to hold it into place. But there's quite often conduction defects associated with using the valve implants. And even with the newer designs, we're still seeing a significant need for permanent pacemaker implantation. So the most common disturbance seen with the valves is left bundle branch block, and also the self-expanding valve type associated with an increased conduction disturbance than the balloon expandable type. And I hope we should be able to see from the next slide that it's put down to just to the size of the actual implant that we're putting in. Because also it's been shown that the more infraannular that a valve is implanted beneath the native aortic valve leaflet, then the increased chance of requiring a pacemaker. So with our models, we were able to potentially segment out the aortic root and have a valve implant, and we can see how it sits in into the valve, and then this can all be incorporated into the whole heart, and we can see how it interacts with the area of the AV conduction axis, and we can take measurements to see potentially what might be impacted on in any way. So there is thought of we could potentially use these models for either improving the design so they don't have the impact on the conduction system, or if there's a valve design in place, then they can be located. We could see potentially how and model how this will impact cardiac conduction and see if there's a better way to go. So hopefully you've seen some of the benefits that micro-CT can offer. It's definitely not intraoperative, but hopefully it offers some kind of value that could potentially lead to some clinical improvements. And it offers high resolution, non-destructive virtual sectioning of tissue, so hearts can be explored very thoroughly, and we're able to identify the cardiac conduction system using the micro-CT. And there's also a large amount of information within the CT data that can be very valuable for computer modelers, which I think could be very important. So the detailed FICTI anatomical models are very valuable educational research tools, and they can help improve our understanding of healthy heart anatomy as well as disease-related modelling. And finally, I'd like to thank particularly Dr. Halina Dobrzynski, who's the head of our lab in Manchester, and lots of students have been involved in the work, and particularly Matthias Holder and Professor Paul Ayazio, who provide much of the heart tissue that we've used in our study. Thank you.